This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of life's deepest mysteries. For decades, we viewed the first cell as a self-contained bubble. But what if the true ancestor of life was something far more fluid? Today, we dive into the radical world of membraneless organelles. We will discover how liquid droplets, not rigid membranes, may have served as the original crucibles of evolution. From the shimmering sheets of mica to the liquid bioreactors still functioning inside our own cells, join us as we rethink the definition of a cell and uncover the fluid origins of life. Chapter 1: The Lipid Divide and the Fluid Ancestor. The story of how life began on Earth is often told as the story of the first cell, and for decades our mental image of that cell has been a tiny, self-contained balloon. In every high school biology textbook, the defining feature of a cell is its membrane, a thin, oily skin that separates the chaos of the outside world from the organized machinery of life inside. This lipid bilayer is seen as the absolute prerequisite for existence, the barrier that keeps precious genetic material and metabolic enzymes from simply drifting away into the vast ancient ocean. However, as we look deeper into the fundamental chemistry of life and the strange structures that still exist inside our own cells today, a different and more radical possibility is emerging. It is possible that the very first functional units of life didn't have membranes at all. Instead, the first precursors to the cell might have been something much more fluid and transient, existing as concentrated droplets of molecular activity that lacked a permanent physical boundary. However, as we look deeper into the fundamental chemistry of life and the strange structures that still exist inside our own cells today, a different and more radical possibility is emerging. It is possible that the very first functional units of life didn't have membranes at all. Instead, the first precursors to the cell might have been something much more fluid and transient, existing as concentrated droplets of molecular activity that lacked a permanent physical boundary. To understand why scientists are questioning the membranes first theory, we must examine the inherent problems with membranes themselves. While they are essential for modern life, membranes are incredibly fragile. They are prone to leaking, swelling, and rupturing under the slightest environmental stress. In the harsh, fluctuating environment of the early Earth, a simple fatty acid bubble would have been a precarious home for the first delicate sparks of metabolism. Modern cells have evolved complex ways to mitigate this fragility. They require robust cell walls in the case of bacteria and archaea, or thick protective protein coats like the pellicle found in paramecium, just to maintain their integrity. A naked prebiotic membrane, lacking these sophisticated reinforcements, would have struggled to survive the physical turbulence of the primordial world. Furthermore, there is a deep evolutionary mystery hidden in the chemistry of the cell itself. If we look at the two most ancient branches of the tree of life, the bacteria and the archaea, we find a startling discrepancy known as the lipid divide. While these two domains share almost all the same internal machinery for reading DNA and synthesizing proteins, their membranes are built from completely different types of fats. Archaea utilize isoprenoid ether-linked lipids, which are exceptionally stable and often branched. In contrast, bacteria and the more complex eukaryotes use fatty acid esters. The biochemical pathways and enzymes required to synthesize these two types of lipids are entirely unrelated and non-homologous. This creates a logical paradox. If a membrane-bound protocell was the last universal common ancestor, LUCA, of all life, it would mean that its descendants evolved two entirely different, complex ways to build a membrane while keeping the internal genetic and metabolic guts exactly the same. This suggests a more plausible alternative. The internal contents of the cell, the genetic and metabolic machinery, reached a high level of complexity before they were ever permanently sealed inside a specific type of lipid boundary. In this view, LUCA was not a discrete membrane-bound cell, but a more fluid entity. This hypothesis is supported by the existence of living fossils within our own cells. We now know that cells organize much of their internal space using membraneless organelles. These structures, often called biomolecular condensates, form through a process where proteins and RNA spontaneously clump together into dense droplets that stay separate from the surrounding watery fluid. While traditional biology has long focused on the roughly 12 types of membrane-bound organelles, such as the nucleus or mitochondria, researchers have now identified a far more expansive landscape of wall-less compartments. Current estimates suggest there are between 30 and 100 distinct types of these membraneless organelles, found in every kingdom of life and managing the most fundamental and ancient processes of the cell. The fact that modern biology relies on such a vast and varied array of fluid assemblies to handle its most critical tasks suggests that they are remnants of a simpler, pre-membrane era of evolution. The transition from chemistry to biology required a way to concentrate the building blocks of life so they could interact. In a vast watery ocean, the laws of thermodynamics and the challenges of chemical concentration make it difficult for complex molecules to form. However, the physical basis for the template of life suggests an early connection to the environment. It is a striking observation that the fundamental periodicity of extended single-stranded nucleic acids and proteins is exactly 0.5 nanometers. This specific atomic spacing is mirrored in certain mineral lattices, suggesting that the first long chains of life were organized by external physical structures before they were ever enclosed in a cell. This model of life's origin fits into a pre-LUCA stage of evolution, where life was not a single discrete organism, but rather a collection of these membraneless units. During this phase, these fluid droplets could have exchanged genetic information and metabolic innovations freely, acting as a sort of global fluid network of biological experimentation. This was a world of liquid complexity, where the essence of life was not the wall that surrounded it, but the dynamic organization of the molecules themselves. The thermodynamic viability of such a system depends on its ability to maintain order against the pressure of entropy. For a prebiotic unit to persist, it must function as a primitive bioreactor, gathering energy and matter to drive its internal reactions. Membraneless droplets offer a unique solution to the chemical challenges of the early Earth, providing a concentrated microenvironment where the water paradox, the difficulty of forming polymers in a watery medium, could be managed. By partially excluding water, these droplets allowed the first metabolic pathways to emerge and become self-sustaining. As these membraneless units became more sophisticated, they would have begun to develop the properties we associate with life, such as growth and division, driven by simple physical forces like shear stress or environmental fluctuations. Selection would have favored droplets that were more efficient at gathering resources or faster at replicating their internal components. Only later, once the internal machinery had become sufficiently complex and the diverse environments of the Earth demanded better protection, did these units begin to wrap themselves in specialized lipid membranes. This perspective shifts our focus from the container to the contents, suggesting that the first functional units of life were defined by their internal logic rather than their boundaries. If the fundamental logic of biological organization is rooted in the spontaneous, self-organizing physics of the molecules themselves, then the first cells were likely not cells at all, but fluid hubs of activity. This leads us to a critical question. How do these wall-less droplets form spontaneously from a simple mixture of molecules? And what physical laws govern their behavior? Chapter 2. Liquid-liquid phase separation and the spontaneous formation of coacervate droplets as primitive protocell models. To understand how the first precursors of life could have functioned without a protective skin, we have to look at a fundamental physical phenomenon that occurs when certain molecules are mixed in water. This process, known as liquid-liquid phase separation, is the secret behind the formation of membraneless organelles. While the term sounds complex, the physics is remarkably intuitive. It is the spontaneous demixing of a uniform solution into two or more distinct liquid phases. Imagine a glass of water into which you pour a small amount of oil. No matter how much you stir, the oil eventually gathers into spherical droplets that remain separate from the water. In the prebiotic world and inside our own cells today, a similar process occurs, but instead of oil and water, it involves highly concentrated clusters of proteins, peptides, and nucleic acids. These clusters are called coacervates, and they represent one of the most compelling models for the very first protocells on Earth. The concept of the coacervate was first championed nearly a century ago by researchers who noticed that when certain oppositely charged polymers are mixed, they don't just sink to the bottom as a solid precipitate. Instead, they form tiny, fluid droplets that stay suspended in the liquid. These researchers suggested that the thick, jelly-like substance of the cell, what was then called protoplasm, behaved exactly like these coacervates. They proposed that life didn't start with a membrane, but with these spontaneous droplets that could gather up organic molecules from the environment and act as a primitive metabolic hub. For decades, this idea was pushed to the sidelines as the focus shifted to the discovery of DNA and the structure of lipid membranes. But recent breakthroughs in microscopy and biophysics have brought the coacervate back to the center of the story. The modern proof for this fluid organization came from the study of P granules, which are small clusters of RNA and protein found in the embryos of microscopic worms. For years, scientists assumed these were solid aggregates, but when they watched them under a high-powered microscope, they saw something extraordinary. When two P granules touched, they didn't just stick together, they fused and merged into a single larger sphere, exactly like two raindrops on a window pane. They also noticed that these granules could deform under pressure and even drip off the surface of a cell's nucleus like liquid. To prove they were truly fluid, researchers used a technique called fluorescence recovery after photobleaching. They hit a small spot on a granule with a powerful laser to bleach the color out of the molecules in that area. Within seconds, the dark spot vanished as unbleached molecules from the rest of the granule flowed into the area. This proved that the interior of these membraneless structures is a highly dynamic liquid environment where molecules are constantly moving and rearranging. The physics driving this formation is a delicate balance of molecular interactions. In a simple solution, molecules are constantly bumping into each other, and the force of entropy usually keeps them mixed and spread out. However, if the attraction between certain molecules is stronger than their attraction to the surrounding water, they will begin to demix. This is often driven by what scientists call multivalent interactions. Think of a molecule as having multiple sticky patches on its surface. If a protein or an RNA strand has many of these patches, it can grab onto several other molecules at once, creating a dense interconnected network. As these networks grow, they eventually reach a critical density where they separate from the bulk fluid, forming a dense phase enriched with these sticky molecules, and a dilute phase where they are scarce. One of the most important versions of this process for the origin of life is complex coacervation. This occurs when two oppositely charged polyelectrolytes, such as a positively charged peptide and a negatively charged strand of RNA, are mixed together. The electrical attraction between the two types of molecules pulls them together, while the release of the small ions and water molecules that were previously surrounding them provides a massive boost in entropy. This thermodynamic win-win makes complex coacervation an incredibly efficient way to create a compartment. In the prebiotic environment, where simple peptides and short strands of RNA were likely abundant, these droplets would have formed spontaneously in any pool of water that reached the right concentration. These coacervate droplets possess several lifelike properties that make them ideal protocell models. First, they are naturally spherical due to surface tension, which minimizes the energy at the boundary between the droplet and the water. Second, they are highly selective about what they let in. Because the interior of a coacervate is a dense, crowded environment with a specific chemical makeup, some molecules will be drawn in while others are pushed out. For example, a coacervate made of peptides and RNA might easily absorb small organic dyes or metabolic building blocks, concentrating them by hundreds or even thousands of times compared to the outside environment. This selective partitioning is the first step toward creating an internal identity that is different from the surroundings, the very definition of a cell. The internal environment of these droplets is also remarkably viscous. Some membraneless organelles, like the nucleolus, have been measured to be 4 orders of magnitude more viscous than water. This high viscosity doesn't just provide stability, it changes the way chemistry happens. In such a crowded space, molecules are forced into close proximity, and the chance of them colliding and reacting increases dramatically. This crowding effect can accelerate enzymatic reactions and help stabilize fragile molecules like RNA hairpins. It also means that these droplets can act as a shield, protecting their contents from being degraded by external factors or fluctuations in the environment. This selective partitioning is the first step toward creating an internal identity that is different from the surroundings, the very definition of a cell. A revolutionary new direction in this research involves active or fuel-driven droplets. Most of the coacervates studied in the past were equilibrium systems, meaning they would form and then stay that way forever unless the temperature or pH changed. But life is never at equilibrium. It is a constant flow of energy. Scientists have now managed to create synthetic coacervates that are powered by chemical fuel. In these systems, a precursor molecule is only able to form a droplet if it is activated by a specific chemical reaction, such as the consumption of a high-energy molecule like ATP or a synthetic equivalent. Once the fuel is used up, the molecule reverts to its inactive state, and the droplet dissolves. These fuel-driven droplets exhibit behaviors that are eerily similar to living cells. They can grow as they consume fuel, and as they reach a certain size, they can undergo spontaneous division. In some experiments, researchers have seen these droplets form internal vacuoles, hollow bubbles of clear fluid inside the dense droplet, which then move to the surface and cause the droplet to break apart into smaller daughter droplets. This process of growth and fission, driven entirely by chemical kinetics and physical forces, provides a plausible bridge to the biological cell cycle. It also solves a major problem in early evolution: how to prevent a droplet from just growing larger and larger until it becomes a single useless mass. By resisting a process called Ostwald ripening, where large droplets grow at the expense of small ones, these active systems can maintain a population of small, dynamic protocells that are better suited for evolution. The chemical diversity of these droplets is another key factor. While many models focus on peptides and RNA, researchers have discovered that even simpler molecules can form these compartments. For instance, alpha-hydroxy acids, which are non-biological molecules found on meteorites and produced in lightning experiments, can polymerize through simple drying and rehydration cycles. These polyesters then spontaneously assemble into microdroplets in water. These polyester droplets can segregate RNA and even support the function of proteins, showing that the ability to form membraneless compartments is a universal property of many different types of polymers. This suggests that the first living droplets might not have been made of the same materials we use today, but could have started with a messy mix of various organic molecules that were available on the early Earth. The stability of these droplets is further enhanced by their ability to interact with salts and minerals. In some systems, certain ions like potassium are preferred over others, and these salts can localize to the surface of the droplet, neutralizing its electrical charge and helping it grow. This interaction with the environment is crucial because it allows the droplet to respond to external cycles, such as the rising and falling of tides or the heating and cooling of a volcanic pool. A droplet that can grow during a period of high salt concentration and then divide during a period of turbulence would be a powerful engine for prebiotic evolution. As these droplets become more complex, they can start to host multi-step chemical reactions. Because they can selectively concentrate different enzymes and substrates, they can act as miniature assembly lines. In one example, researchers created a coacervate that could capture two different molecules and facilitate a reaction between them that was nearly impossible in the open water. By stabilizing intermediate states that would normally fall apart, the droplet allows for the buildup of complex organic structures. This ability to act as a chemical crucible is what would have allowed the first metabolic pathways to emerge and become self-sustaining. The transition from a simple droplet to a complex protocell also involves the way these structures handle information. While a coacervate is a liquid, it is not a chaotic one. The sticker spacer model helps explain how these droplets maintain their organization. In this model, the stickers are the parts of a molecule that provide the attractive force, while the spacers provide the flexibility and the volume. By changing the sequence and spacing of these components, the properties of the droplet, such as its viscosity, its surface tension, and what it lets inside, can be precisely tuned. This means that even a simple peptide sequence can encode the physical properties of the compartment it builds. This coupling between molecular sequence and physical structure is a primitive form of the relationship between a genotype and a phenotype. In the early Earth, a population of these droplets would have been a massive natural experiment. Some droplets would have been better at gathering fuel, some would have been more stable, and some would have been faster at replicating their internal RNA. Because these droplets can exchange materials by fusing and breaking apart, they could share their innovations with each other. This fluid network of competing and cooperating droplets would have been the perfect environment for the first sparks of natural selection to take hold. The droplets that were most efficient at managing their internal chemistry would have persisted longer and produced more daughter droplets, gradually leading to the emergence of the sophisticated molecular machinery that defines life. The discovery that these fluid structures can also form in prokaryotic cells, which were long thought to be simple bags of enzymes, has been a major revelation. It shows that even the most ancient and simple organisms on Earth still rely on the same physical principles of phase separation to organize their internal life. We see this in specialized membraneless structures like carboxysomes, which bacteria use to concentrate the enzymes needed for metabolic pathways. What is particularly striking is that these organisms achieve this complex organization despite the fact that only about 6% of their proteins are intrinsically disordered, a much lower proportion than in more complex cells. This ubiquity across all branches of life is a powerful indicator that the liquid-liquid phase separation we see today is not a recent evolutionary invention, but a fundamental legacy of the very first steps life took on this planet. These droplets also provide a unique solution to the problem of how the first genetic material was protected. In the open ocean, a strand of RNA is vulnerable to being broken apart by water or ultraviolet light. But inside a dense coacervate, the RNA is surrounded by a protective cloud of peptides and other molecules. This environment can even help the RNA fold into functional shapes, like ribozymes, which can then start to catalyze their own replication. The droplet doesn't just hold the RNA, it provides the specific chemical microenvironment, the right pH, the right salt concentration, and the right molecular crowding that the RNA needs to be active. This fluid foundation for life's origin shifts our understanding of the first cell from a static object to a dynamic process. It suggests that the first units of life were not defined by their boundaries, but by the internal logic of their molecular interactions. The coacervate droplet is a self-organizing system that emerges naturally from the laws of physics and chemistry, creating a pocket of order in a disordered world. This liquid complexity provided the essential starting point for the long journey toward the first membrane-bound cells, acting as a flexible and resilient nursery for the molecular machinery that would eventually build the world we know. As these fluid assemblies became more stable and specialized, they began to incorporate more complex molecules, leading to the formation of the first organized ribonucleoprotein complexes. Chapter 3. The role of intrinsically disordered proteins and RNA in nucleating ancient ribonucleoprotein assemblies. If we were to zoom in past the shimmering surface of a coacervate droplet and look directly at the molecular machinery inside, we would find a world that looks very different from the rigid, highly structured diagrams found in most biology textbooks. In a modern cell, we are often taught that proteins are like precision-engineered parts, each folded into a perfect three-dimensional shape that allows it to function like a key in a lock. But the proteins that build membraneless organelles, and the ones that likely dominated the prebiotic world, do not follow these rules. They belong to a class of molecules known as intrinsically disordered proteins. These molecules are floppy, flexible, and constantly shifting, more like pieces of wet string than stiff gears. It is this very lack of structure that made them the perfect partners for RNA in the ancient dance that led to the first functional units of life. To understand why these disordered proteins were so vital, we have to look at their chemical signatures. They are characterized by what scientists call low sequence complexity. While a modern, highly evolved enzyme might use a precise arrangement of all 20 different amino acids to achieve its shape, disordered proteins often rely on a limited palette of simple, repeating amino acids. In the messy environment of the early Earth, these simple sequences would have been much easier to form spontaneously through abiotic chemistry. It is far more plausible for a chain of a few repeating amino acids to emerge from a mineral crevice or a volcanic pool than for a perfectly folded 300-link protein to appear by chance. These simple, flexible chains were the first scaffolds of life, using their floppiness as a strength rather than a weakness. This process of nucleation is one of the most important steps in the origin of life. In a vast primordial soup, the building blocks of life would have been spread thin. Without a way to gather them together, chemical reactions would happen too slowly to sustain a living system. RNA strands provided the physical platform for this gathering. A single long molecule of RNA can grab on to dozens of disordered proteins at once, pulling them out of the surrounding water and into a dense, localized hub. This doesn't just create a compartment, it creates a reaction center. By holding the proteins in close proximity, the RNA allows them to interact, fold, and eventually begin to catalyze chemical reactions. The most ancient and important example of this ribonucleoprotein partnership is the ribosome. Found in every living thing on Earth, the ribosome is the massive molecular machine that builds proteins by reading genetic code. Despite its complexity, the core of the ribosome is a dense cluster of RNA and protein that has remained virtually unchanged for billions of years. It is, in effect, a small, highly specialized membraneless assembly that eventually became so stable it was frozen in time. The fact that the most fundamental process of life, the creation of proteins, is managed by an RNP complex is a powerful clue. It suggests that before there were cells, there were these small RNP assemblies, and that they likely began as fluid, membraneless droplets that gradually became more structured and precise. We can see the legacy of this fluid beginning in the way modern cells build their ribosomes. Inside the nucleus of a eukaryotic cell lies the nucleolus, a large wall-less droplet that functions as a ribosome factory. The nucleolus is a masterpiece of liquid-liquid phase separation, organized into layers like an onion. Each layer has a different viscosity and a different chemical makeup, allowing the various stages of ribosome assembly to happen in a specific order. The fact that the cell still uses a membraneless droplet to house its most ancient manufacturing process suggests that this was the original way life was organized. The nucleolus is a living window into a time when life was a collection of fluid, nested compartments, each one a specialized hub for a different part of the metabolic puzzle. The specific shapes that RNA takes also play a role in how these assemblies form. While DNA is usually a long, straight double helix, RNA is much more adventurous. It folds back on itself to form intricate shapes like hairpins, loops, and junctions. We now know that these structured regions of RNA are exceptionally good at partitioning into membraneless droplets. A base-paired RNA hairpin, for instance, will be drawn into a coacervate much more effectively than a random unstructured strand of the same length. This means that as RNA molecules evolved more complex shapes, they also became better at organizing the droplets they lived in. This created a feedback loop. More complex RNA led to more stable and specialized droplets, which in turn provided a better environment for the RNA to replicate and evolve further. The proteins involved in these ancient assemblies also show a preference for simplicity in their internal structures. In many ribonucleoprotein particles, the proteins utilize crossed beta structures. These are simple, repeating patterns where the backbone of the protein is stretched out rather than wound into a tight spiral like an alpha helix. Beta structures are much easier to form from simple amino acid sequences and are more robust in the face of environmental stress. They provide a sturdy but flexible framework that can support the RNA core. This suggests a progression where the very first proteins were almost entirely disordered, and then gradually developed these simple beta structures to provide more stability to the RNP hub. One of the most fascinating aspects of these disordered proteins is their ability to respond to environmental cues. Because they lack a rigid shape, they are highly sensitive to changes in salt concentration, pH, and temperature. A small shift in the acidity of a pool of water could cause a disordered protein to suddenly change its stickiness, causing a membraneless organelle to form or dissolve in seconds. This provided early life with a built-in sensing system. These fluid assemblies didn't need a brain or a nervous system to respond to their world. Their very physical structure was a sensor. This allowed the first protocells to adapt to the fluctuating conditions of the early Earth, gathering their components together when resources were available and dispersing them when conditions became too harsh. We see a modern echo of this adaptive sensing in the formation of stress granules. These are a specific type of membraneless organelle that cells assemble rapidly to store mRNA and temporarily halt the production of unnecessary proteins during periods of cellular stress. By sequestering these blueprints, the cell can focus its metabolic energy on survival until the environment stabilizes. This modern mechanism is a direct descendant of the ancient ability of disordered proteins to condense and protect vital genetic material when the prebiotic world turned hostile. This sensitivity also leads to a phenomenon known as molecular aging or maturation. Over time, a fluid droplet can become more viscous, eventually transitioning into a gel or even a solid-like state. While this can be a problem in modern biology, where the failure of proper phase separation is a primary factor in neurodegenerative conditions such as ALS, Alzheimer's, and Huntington's disease, in the prebiotic world, it might have been a vital tool for evolution. A fluid droplet that could ripen into a more stable gel would have been better protected from being washed away. This maturation process represents the first steps toward building the more permanent, structured organelles we see in cells today. It is the transition from a transient, temporary gathering of molecules to a stable, long-lived biological structure. The chemical motifs found in these disordered proteins are also remarkably consistent across the tree of life. Many of them contain high concentrations of the amino acid glycine, which provides extreme flexibility, and aromatic amino acids like phenylalanine or tyrosine, which act as the stickers that provide the attractive force. Others rely on blocks of alternating charge, stretches of positive amino acids followed by stretches of negative ones. These charged blocks allow the proteins to interact with each other and with RNA through long-range electrostatic forces. These same motifs are found in the proteins that manage RNA in your own cells right now, in the proteins that form the pores of your cell nucleus, and in the proteins that help your neurons form long-term memories. They are a universal molecular language for organizing life without membranes. The partnership between RNA and disordered proteins also solved the problem of how to handle the messy chemistry of the early Earth. The primordial soup was not a clean laboratory. It was filled with a huge variety of organic molecules, many of which were useless or even toxic to the first sparks of life. Membraneless droplets provided a way to filter this mess. Because they are highly selective, they could gather up the good molecules, the nucleotides and amino acids needed for life, while excluding the bad ones. The disordered proteins acted as a sort of molecular sieve, creating a clean, concentrated microenvironment where the delicate work of building the first genetic chains could happen without interference. As these RNP assemblies became more sophisticated, they would have started to develop a division of labor. One droplet might become specialized in replicating RNA, while another might focus on building simple peptides. Because these droplets can easily fuse and exchange materials, they could have functioned as a global network of chemical innovation. This is the RNA world in its most plausible form. Not just strands of RNA floating in an empty ocean, but a world of fluid, nested RNP hubs, each one a tiny laboratory for a different part of the life-making process. This fluid organization provided the perfect balance of stability and flexibility, allowing the first functional units of life to emerge and evolve in a world that was constantly changing. The transition from these fluid hubs to more permanent structures was likely driven by the proteins themselves. As disordered proteins began to incorporate more stickers and develop more structured regions, the droplets they built became tougher and more resilient. Some might have even begun to form a primitive sort of skin, not out of lipids, but out of a dense layer of protein and RNA. This would have provided a new level of protection, allowing the RNP hubs to survive in even more challenging environments. This gradual hardening and specialization of the fluid droplet is the story of how the first true organelles were born, setting the stage for the eventual arrival of the lipid membranes that would define the modern cell. This perspective shifts our focus from the history of the cell wall to the history of the cell's contents. It suggests that the most ancient parts of our biology are not the containers, but the fluid, disordered networks of RNA and protein that still manage our most fundamental processes. Our own cells are still, at their heart, organized by the same liquid-liquid phase separation that governed the very first RNP assemblies on the early Earth. We carry within us a legacy of that liquid world, a world where life began not as a rigid structure, but as a flexible, adaptive, and highly creative partnership between two of the most versatile molecules in the universe. This ancient partnership provided the foundation for everything that followed, creating the first pockets of order in a disordered world and paving the way for the emergence of the first true cells within the unique geological niches of the ancient Earth. Chapter 4. Geological niches for prebiotic compartmentalization, including the spaces between mica sheets and mineral pores. While the fluid dynamics of membraneless droplets provide a compelling logic for how the first functional units of life could have organized their internal chemistry, a critical question remains. Where did these droplets actually exist? In the vast, turbulent oceans of the early Earth, a tiny cluster of RNA and protein would have been a needle in a haystack, constantly threatened by dilution and the harsh conditions of the open environment. To survive long enough to evolve, these fluid assemblies needed more than just internal logic. They needed a physical home, a geological nursery that could provide shelter, concentrated resources, and a steady supply of energy. Recent scientific inquiry has turned its gaze away from the open sea and toward the rugged, porous landscapes of the Earth's crust, identifying specific mineral environments that could have acted as the ultimate cradles for the emergence of life. One of the most remarkable candidates for this geological nursery is a mineral that many of us are familiar with in everyday life, mica. Specifically, muscovite mica, with its shimmering, layered sheets, possesses a set of physical and chemical properties that are almost eerily well suited for the origin of life. Mica is a silicate mineral that forms in thin, flexible layers, and it is the environment between these layers that holds the secret. These spaces are not empty. They are filled with water and bridged by a high concentration of potassium ions. This is a significant detail because every living cell on Earth today maintains a high internal concentration of potassium, while the surrounding ocean is dominated by sodium. Most origin of life theories struggle to explain how or why the first cells would have developed a preference for potassium. But the mica hypothesis provides a simple answer. Life began in a potassium-rich environment, and our modern cellular chemistry is a direct legacy of those ancient mineral gaps. The physical structure of the mica surface itself offers another profound clue. The crystal lattice of mica is arranged in a hexagonal grid of atoms with a periodicity of exactly 0.5 nanometers. This specific measurement is not a coincidence. It is the same spacing found between the building blocks of extended single-stranded nucleic acids and carbohydrate polymers. This means that the mica surface could have acted as a physical template, a sort of molecular workbench where the first long chains of RNA were held in place and organized. Unlike other minerals like clay, which swell and shrink violently when they get wet or dry, mica is much more stable. It allows water to move in and out between its sheets in a gentle, controlled process, providing a sheltered environment where the thickness of the water layer can vary from the sub-nanometer scale to several microns. This range of scales is perfect for hosting everything from individual molecules to the largest membraneless organelles. Perhaps the most revolutionary aspect of the mica hypothesis is the role of mechanical energy. We often think of the energy for life's origins coming from chemical gradients or the sun, but mica provides an entirely different source, the movement of the Earth itself. Because mica sheets are thin and flexible, they can move apart and together in response to external forces, like water currents or temperature fluctuations. This open and shut motion creates a mica spring that can generate a wide range of mechanical energies. This isn't just passive movement, it is a form of work that can be used to drive chemical reactions. This process, known as mechanochemistry, occurs when physical force is used to push molecules into close proximity, overcoming the energy barriers that would normally prevent them from bonding. Imagine a simple amino acid like alanine trapped between two moving mica sheets. As the sheets are pushed together by a passing wave or a change in temperature, they physically squeeze the alanine molecules, forcing them to react and form a peptide bond. This could have been the original way that long protein chains were built, long before the first enzymes ever existed. In this model, the moving mica sheets functioned as the first enzymes, using physical motion to catalyze the formation of chemical bonds. There is strong evidence for this in modern biology, where we see that enzymes like hexokinase or lysozyme undergo large physical motions, opening and shutting like a jaw, to bring their substrates together and trigger a reaction. The mechanical energy of the Earth, harnessed by the flexible sheets of mica, could have provided an endless supply of energy to drive the transition from non-living matter to the first complex polymers. While mica provides a specialized environment for mechanochemistry, other researchers are looking at a broader geological context, the early continental crust. It is now believed that the continental crust began to form very early in Earth's history, shortly after the surface cooled. This crust was likely riddled with tectonic rift zones and fault zones that remained active for tens of millions of years. These zones were filled with water and gases, creating a diverse and highly pressurized environment that functioned as a massive, planet-scale chemical reactor. In these deep faults, the Earth provided everything needed for life: an abundant supply of raw materials, cyclic fluctuations in pressure and temperature, and a variety of metallic catalysts. The conditions within these crustal faults were far more complex and productive than the warm little pond models of the past. High-pressure laboratory experiments have shown that these environments can facilitate the transition of gases between supercritical and subcritical phases, a process that is incredibly efficient at concentrating organic molecules. Fluids trapped within these faults often have a very low pH, making them acidic. This is another vital detail, as RNA is most stable at a pH of 4 to 5. In the alkaline environment of the open ocean, RNA would have broken down quickly, but within the acidic crevices of the continental crust, it could have persisted and grown.
The chemical diversity found in these mineral niches is staggering. Analysis of fluid inclusions in ancient hydrothermal minerals, such as quartz and calcite, has revealed a wide variety of organic molecules, including aldehydes, which are essential precursors for lipids. These fault zones also would have been rich in sulfide ores. These ores often contain radioactive elements that could have provided a steady source of energy to drive early reaction networks. Furthermore, the cations released from the surrounding rocks as they were altered by the acidic, high-pressure fluids would have acted as natural catalysts, facilitating the synthesis of the building blocks of life. Recent studies have demonstrated that at least 12 different amino acids, including glycine, alanine, and valine, can form spontaneously under the conditions found in these hydrothermal faults. Even more impressively, all 5 nucleobases, the structural components of DNA and RNA, as well as ribose and deoxyribose sugars, have been synthesized in laboratory simulations of these environments. This suggests that the early continental crust was not just a shelter, but a factory, constantly churning out the raw materials needed to build the first membraneless organelles and protocells.
The porous nature of the rocks in these rift zones provided a tiered form of compartmentalization. At the macro scale, the mineral pores themselves acted as the first containers, preventing the precious organic soup from being lost. Within these pores, the physical environment, the temperature gradients, the pressure cycles, and the specific mineral surfaces would have driven the molecules to organize. This is where liquid-liquid phase separation would have taken place. The mineral pore provided the outer boundary, while the spontaneous formation of coacervates and other membraneless droplets provided the internal organization. This tiered structure solves one of the biggest problems in the origin of life: how to create a complex, organized system without starting with a pre-made cell wall. These geological niches also provided a solution to the water paradox. Many of the reactions needed to build life's polymers, like the formation of peptides or RNA chains, are dehydration reactions. They release a molecule of water. In a vast watery ocean, these reactions are thermodynamically unfavorable. However, in a mineral pore that undergoes regular cycles of drying and rehydration, or in a deep fault where supercritical fluids are present, the local environment can be manipulated to favor these reactions. The mineral surfaces can adsorb water molecules, pulling them away from the reaction site and allowing the polymers to grow. This suggests that life didn't emerge in a static environment, but in one that was constantly fluctuating, using the cycles of the Earth to drive its internal chemistry forward. The interaction between these geological niches and early membraneless assemblies would have been a dynamic partnership. For example, the vesicle membranes that eventually became the walls of the first cells might have first formed as a reservoir for hydrophobic amino acids within a mineral crevice. These membranes could have then facilitated the attachment of those amino acids to proto-RNA molecules. In the early continental crust model, it is hypothesized that proto-tRNA served a dual role during this stage, functioning as both a transfer agent for amino acids and a template for encoding their sequences. In this scenario, the geological niche provides the physical space and the energy, the mineral surfaces provide the templates and catalysts, and the fluid droplets provide the concentrated reaction centers. This multi-layered approach to compartmentalization creates a much more robust and plausible pathway for the transition from simple chemicals to the first living systems. The role of shearing forces within these mineral environments is also a key factor in early evolution. As fluids were forced through narrow cracks in the crust, or as mica sheets moved against each other, they would have created significant physical stress. For a simple membraneless droplet or a primitive vesicle, these shearing forces could have been the original mechanism for division. Instead of needing a complex set of proteins to coordinate cell division, the first protocells could have been physically broken apart into daughter units by the movement of their environment. This represents a pre-LUCA stage of evolution, where the fundamental processes of growth and reproduction were driven by the physics and geology of the Earth itself. This perspective also highlights the importance of the specific mineralogy of the early Earth. The presence of sulfide ores, carbonates, and arsenites on the walls of tectonic faults would have created a variety of reactive surfaces, each favoring a different type of chemistry. Some surfaces might have been better at building peptides, while others were better at stabilizing RNA. Because these faults were interconnected networks of pores and cracks, the products of these different chemical laboratories could have been mixed and matched, allowing for a level of combinatorial complexity that would be impossible in a single uniform environment. This fluid interconnected network of mineral niches was the ultimate platform for biological experimentation. The stability of RNA within these environments is particularly noteworthy. While RNA is often seen as a fragile molecule, its stability is highly dependent on its surroundings. In the presence of certain divalent cations, like magnesium, which would have been abundant in these mineral-rich fluids, RNA can fold into complex, functional shapes called ribozymes. These ribozymes could then start to catalyze their own replication. However, this creates a significant challenge for early membrane models. While magnesium is required for the activity of primitive RNA catalysts, fatty acid vesicles are notoriously unstable in the presence of these same ions. The mineral pores provided the perfect reaction vessel to resolve this conflict, concentrating the RNA and its magnesium cofactors while protecting the fragile catalysts from degradation within a wall-less compartment that did not rely on unstable lipids. The acidic nature of these fluids would have further enhanced this stability, ensuring that the first genetic information was preserved long enough to be passed on to the next generation of droplets. As we look at the potential for life on other planets, this geological perspective becomes even more important. If life's origin is tied to the specific physics of mica sheets or the high-pressure environments of continental rift zones, then we should be looking for these same features on other worlds. Rocky planets with active tectonics, layered minerals, and hydrothermal systems become the primary targets in our search for extraterrestrial life. The mica nursery and the crustal reactor are not just stories about Earth's past. They are universal models for how a planet can harvest its own mechanical and thermal energy to spark the transition from chemistry to biology. The transition from these geological nurseries to the first free-living cells was likely a long and gradual process. At some point, the internal machinery of the membraneless organelles became sophisticated enough to synthesize the lipids and proteins needed to build a permanent, self-sustaining membrane. This would have allowed the first cells to finally leave the safety of the mica sheets and the mineral pores, carrying their potassium-rich internal environment with them as a permanent reminder of their mineral origins. This move from the geological to the biological was the defining moment in the history of our planet. But it was only made possible by the unique physical and chemical advantages provided by the Earth's own crust. The discovery that even simple bacterial cells today still utilize these same principles of compartmentalization is a powerful confirmation of this theory. Bacteria use membraneless droplets to organize their RNA and to concentrate the enzymes needed for metabolic pathways, showing that the fundamental logic of life's organization has remained unchanged for billions of years. We are not just biological entities. We are the descendants of a liquid-mineral partnership that began in the dark, pressurized crevices of a young planet. This ancient partnership provided the stability, the energy, and the organizational framework that allowed the first fluid droplets to function as true bioreactors.
Chapter 5. The Liquid Bioreactor. The transition from a chaotic mixture of simple organic molecules to the first self-sustaining systems of life required more than just a physical enclosure. It required a highly efficient chemical engine. In the vast, dilute environment of the early Earth, the chances of the necessary building blocks, nucleotides, amino acids, and lipids, randomly bumping into one another in the right orientation and at the right time were vanishingly small. To overcome this hurdle, the first membraneless droplets functioned as sophisticated bioreactors. These fluid crucibles didn't just hold molecules. They actively gathered, sorted, and concentrated them, creating a chemically distinct biophase, where absorbed water possessed different solvent properties than the bulk ocean. This functional capacity to act as a reaction hub is what allowed these droplets to drive the first stages of prebiotic evolution, turning a messy soup into an organized network of reactions. The primary power of the membraneless droplet lies in its ability to selectively sequester materials from its surroundings, a process governed by the Meyer-Overton rule. This physical law dictates how small molecules partition into the biophase based on their solubility, ensuring the droplet's interior was not merely a concentrated version of the outside water. Because the interior of a coacervate or a polyester droplet is a dense, chemically distinct phase, it acts as a molecular sponge. Negatively charged compounds and positively charged molecules can be drawn into these droplets, reaching concentrations hundreds of times higher than in the outside water. Even neutral molecules are captured if the droplet contains hydrophobic microdomains. This ability to recruit diverse substances simultaneously allowed a single droplet to gather the ingredients needed for both metabolism and the eventual synthesis of membranes. This concentration effect solves the problem of molecular crowding, which is essential for life. By forcing molecules into close proximity, the droplets dramatically increase the frequency of molecular collisions. This crowding doesn't just speed up reactions, it changes the thermodynamics of the system. In a crowded environment, the effective volume available to a molecule is reduced, which can push proteins and RNA to fold into their functional shapes and stabilize fragile structures that might otherwise fall apart in a more dilute setting. While the open ocean drove hydrolysis, the droplet's internal water activity was suppressed, allowing dehydration reactions to proceed in a dry within wet microenvironment. The chemical work done inside these droplets goes far beyond simple concentration. They function as catalysts that organize complex pathways to abolish unwanted side reactions. Much like modern synthetic RNA-based organelles, early droplets could sort messy chemistry, ensuring that metabolic intermediates were funneled toward productive ends rather than being lost to chaotic cross-reactions. One striking example of this is the aldol condensation reaction, a fundamental process for building complex carbon chains. In the open water, this reaction is slow and inefficient, but inside simple peptide coacervates, the rate of reaction is significantly enhanced. The droplet acts as a scaffold, holding the reactants in the correct orientation and stabilizing the transition states that occur during the reaction. Within the dense interior of these droplets, genetic complexity was driven by structural organization. DNA molecules can organize into liquid crystals within the coacervate phase, a structural alignment that facilitates non-enzymatic ligation. This allows shorter genetic fragments to link into longer, more complex chains without the need for advanced biological machinery. This is a critical feature, as primitive coacervates were likely formed by much shorter peptides than those found in modern biology, a constraint imposed by the lack of sophisticated translation systems. These short-chain assemblies provided the necessary stability to capture and shield fragile intermediate molecules, such as imines, allowing multi-step reaction networks to build up into a primitive metabolism. The functional complexity of these droplets is further demonstrated by their ability to process information through tiered compartmentalization.
Researchers have used these systems to create Boolean logic gates that can sense specific chemical or physical inputs, such as a change in light or the presence of a particular molecule, and produce a defined output. For instance, a droplet might be designed to only assemble or release a cargo if both a certain pH level and a specific salt concentration are met, effectively acting as an AND gate. This capacity for signal processing suggests that early protocells could have responded to their environment in a way that mimics the behavior of living organisms, moving beyond passive chemistry toward active computation. To understand how these functional bioreactors evolved, we must look at the concept of thermodynamic selection. In the prebiotic world, selection started with persistence, described by a reaction viability inequality. For a droplet to persist, the energy it gathers from the environment, plus the energy it releases during its internal reactions and the energy it has stored, must exceed the energy it spends to maintain its structure. Droplets that were more efficient at gathering high-energy molecules or better at recycling their internal components would have been more viable. They would have lasted longer and had more opportunities to grow, while less efficient droplets dissolved back into the environment.
This persistence-driven selection is amplified in active systems that can resist Ostwald ripening. In a normal mixture, large droplets tend to grow while small ones shrink and vanish, eventually leading to a single inert mass. However, active droplets maintain a steady population of small, uniform units. This is vital for evolution because it ensures a large population of individual protocells that can compete and vary. The physical fitness of a droplet was determined by the specific sequence of its components. A mutation that altered the arrangement of attractive patches could transform a fluid hub into a more robust or catalytic engine. Even before a complex genetic code, there was a direct link between a molecule's sequence and the physical fitness of the compartment it created. As these droplets evolved, they began to exhibit increasingly complex social behaviors, such as communication and predation. In synthetic models, researchers have observed predatory behavior, where one type of droplet can physically engulf another to harvest its resources. Other systems show communication, where a chemical signal released by one droplet triggers a change in the composition of a neighbor. This suggests that the early Earth was filled with complex communities of interacting membraneless units. This social environment would have accelerated evolution by allowing for the rapid exchange of chemical innovations and the development of specialized roles within a group. Environmental energy sources, such as external electric fields, could drive the fission of these droplets, providing a physical mechanism for reproduction. These fields drive the release of daughter droplets, ensuring the continuation of successful molecular lineages. The role of metal ions and salts in these processes is another key driver of complexity. Droplets manage these ions selectively, preferring potassium over sodium to neutralize surface charges, or using magnesium as a vital cofactor for catalytic reactions. This selective ion management is one of the most ancient functions of the cell, beginning within these first fluid bioreactors. The transition from these fluid droplets to the first true cells involved a process of maturation. Over time, a droplet that was highly successful at building complex polymers might have become more viscous, eventually forming a stable gel-like core that provided a permanent architecture for metabolic machinery. Around this core, lipids and other amphiphilic molecules, including those formed in polyester droplets, would have naturally gathered. While it was once thought that membranes required existing membranes to grow, recent experimental evidence demonstrates that lipid bilayers can form de novo without pre-existing templates or protein machinery. This spontaneous assembly provides a potential solution to the paradox of membrane heredity, allowing a fluid bioreactor to wrap itself in a permanent skin once its internal chemistry reached a critical threshold. The droplet provided the template and the concentrated building blocks, while the membrane provided the final layer of protection and control. The legacy of these early bioreactors is still visible in the way modern cells handle their most critical tasks, from the formation of stress response granules to the specialized factories that build ribosomes. These modern organelles are not just coincidental structures. They are highly evolved versions of the very first bioreactors that sparked life into existence. They remind us that the fundamental unit of biological organization is not a rigid wall, but a fluid, dynamic space where chemistry is concentrated and controlled. If the first steps of life were taken within these wall-less liquid crucibles, how did the eventual arrival of a permanent lipid membrane change the trajectory of evolution from a communal fluid network to the discrete, competing individuals we recognize as cells today? The journey from primordial chaos to complex life was not a sudden leap into a sealed container, but a gradual evolution of liquid complexity. We have explored how membraneless organelles, driven by the physics of phase separation and the flexibility of disordered proteins, provided the first functional hubs for metabolism and genetic replication. These fluid bioreactors, sheltered within mineral niches like mica, allowed life's internal logic to mature long before the first lipid membranes were ever forged. This shift in perspective reveals that the essence of life lies not in the walls that divide us, but in the dynamic, self-organizing chemistry within. As we look at the membraneless structures still operating inside our own cells, we see a living legacy of Earth's first fluid ancestors. Perhaps life is less a thing we possess and more a liquid process we inhabit. Thank you for listening.